In the early 20th century, most biologists believed that protein acted as a genetic material. Following the work of Avery, McLeod, and McCarty in 1944, there was emerging evidence that DNA was the molecular basis for inheritance, but their work was not viewed as conclusive evidence. Conclusive evidence was not introduced until 1952 when Alfred Hershey and Martha Chase set out to study T2 bacteriophage, which is a phage or virus that infects bacteria. It was known that T2 bacteriophage was composed of proteins and DNA. They also knew that when it infects bacteria, it injects something inside that hijacks the bacteria's genetic information to start producing more of the T2 bacteriophage. If they could show what that something was, then they could prove that it was either protein or DNA that forms the molecular basis. They developed an experiment to figure out what exactly the T2 bacteriophage was injecting. They created 2 batches of T2 bacteriophage. Batch 1 was created in the presence of radioactive phosphorus. The other was created in the presence of radioactive sulfur. These isotopes were chosen because phosphorus is found in DNA and would therefore incorporate itself into the DNA. Sulfur is found in protein and would therefore incorporate itself into the protein. These radioactive batches were created by growing the viruses in either the radioactive sulfur or radioactive phosphorus medium. As they reproduce, they incorporate the radioactive material in either the protein or the DNA of the new viruses that get produced. The bacteriophages created were then allowed to infect a bacteria. Once infected, they used a blender to detach the remaining bacteriophage from the outside of the bacteria. The samples were then put into a centrifuge to separate the supernatant from the pellet. In a centrifuge, the heavier material will gravitate to the bottom of the tube and the lighter to the top. Bacterial cells are heavier and will fall to the bottom and create a pellet. The leftover fluid and phage parts are lighter and will gravitate to the top. If the pellet collects phosphorus, that means that the DNA made it into the bacteria, but if the sulfur is found in the pellet, it means that the protein made it into the bacteria. At the conclusion of the experiment, Hershey and Chase checked each pellet for the presence of radioactivity and found that the radioactive phosphorus was in the pellet. They were able to conclude that it was DNA that made it inside of the bacteria and not the protein. Their experiment proved Avery, McLeod, and McCarty correct. The DNA is the genetic material.